Hello, everyone, and welcome to today's webinar titled Human-Induced Pluripotent Stem Cell Expansion in a Stirred 3D System, an example of manufacturing allogeneic resources for cell therapy. This webinar is a part of the 5th annual event in the Bioprocessing Virtual Event Series. I am Shelley Mulock of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by DASGIP, an Eppendorf company. For more information about DASGIP, an Eppendorf company, go to eppendorf.com, EPPENDORF.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them in the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain your credits. I now present today's speaker. Dr. Jorge Escobar is a senior research scientist in the Applications Lab of Eppendorf Incorporated. For a complete biography on our speaker, please visit the presenter tab from the menu at the left of your screen. Dr. Escobar, you may now begin your presentation.
Thank you for this kind introduction. It's a great pleasure for me today to talk to the audience and explain the platform we have developed for human-induced pluripotent stem cells and expansion in a stirred 3D system as a perfect example for allogeneic cell therapy applications. So Eppendorf is a leading life science company founded in Hamburg, Germany in 1945 with the purpose of improving human living conditions. So our company has subsidiaries in over 26 countries, developing and providing solutions to our customers with the mission to bring life-saving treatments to the world. So the application labs located in Enfield, Connecticut, and Jülich in Germany, along with the Eppendorf Application Technology Lab located in Namur in Belgium, develop bioprocess solutions for our customers in cell and gene therapy, proteins, and vaccine production. Let me see. So in this regard, our colleagues or our researchers have developed over the years an extensive library of application notes with specific details about vaccines and protein productions and cell and gene therapy workflow, like the one I'm planning to present to you today. So going more into details, let me talk a little bit more about the advanced therapy. So advanced therapy medicinal products, ATMP, are innovative biopharmaceutical for human use, where the active substance is the biological substance or is harvested from a biological source. So as you see in the slide, US and EU have different regulation, but they have similar subclassification like gene therapy and cell therapy. And in the case of Europe, they have two more subclassification as tissue engineered products and also the combination of a TMP product that could be, for example, a medical device like cells embedded in a matrix or a scaffold for tissue regeneration. By looking at the complexity and size, you can see that advanced therapies are much more complex than the biologics or small molecules, which means the ATMP are high-tech products developed on the very specific, complicated process that are at the forefront of innovation with more chances of finding cure for patients. So the aim between the small molecules and the advanced therapy is quite different. I have to say that. And it is not to have a pharmacological effect in the body. The idea is basically to introduce cells or tissue in the human body, even in the site of the defect of the disease or remove from there in order to replace or restore lost functions, or even introduce genetic material into the cells or tissue to compensate for its absence. So it's important to highlight here that this is a revolutionary technology where the cells are living drugs and with the ability to respond to the microenvironment in a very personalized way. So talking more specifically about cell and gene therapy, CGT, this is an important part of the ATMP, and they are a transformative new category of medicines, and their full potential is just beginning to start. So cell and gene therapies involve extracting cells, as you see in the slide, proteins or genetic material from the patient or the donor, and engineering them to provide a highly personalized therapy where the cells or this genetic information is re-injected or reinfused into the patient. In addition to that, I would like to say that they have the potential to address a complex diseases and rare disorder for which there is no effective cure yet. Talking a little bit about the market, you see that the global cell and gene therapy market is set to grow close to $10 billion from 2022 to 2026, according to Technoview. And the market growth momentum will accelerate with the growth around 20%, with the cell therapy segment being the largest contributor to the market, and North America accounting for 41% of the markets over the forecast period. So there are different types of cell and gene therapy approaches, and autologous is one of them. And I have to say that this is really an example of a personalized medicine. So cells are extracted from the patient and infused again into the same patient after a laborious procedure of cell isolation, expansion, and validation. So as other autologous cell therapy strategy present key features and challenge. And for example, the cell therapy has very low immunogenicity. That means that the cells are from the same patient and they have demonstrated strong efficacy in different clinical trials. However, the strategy presents also several challenges. For example, manufacturing, and that can take 2 to 3 weeks to make a therapeutic dose while patients may progress waiting for the treatment. The cost is high also, and the laws are personalized and manufacturing process are difficult to scale up effectively. And finally, as you see in the slide, they have poor reliability, meaning that if something goes wrong, during the manufacturing, the patient will not receive the therapy on time. Regarding to that, I mean, this challenge raised questions about whether an alternative approach can be considered, taking into consideration of this all this knowledge accumulated with the current autologous strategy, and offer a more simple solution to develop a cell therapy product for many patients. So this is a case of the allogeneic approach, where cells from a healthy donor and are collect, expand, and used for many patients. From the manufacturing point of view, the goal is to develop a scale-up model capable to produce affordable batches for 100 patients. And this is, I want to say that this is a really truly off-the-shelf product. So I want to show here a clear example of allogeneic strategy, specifically an allogeneic CAR T cell therapy approach. I don't want to go more really into detail here, but as you can see in the slide, there are different steps in the upstream, such as T cell isolation, the preculture, the T cell characterization and banking, going into the scale-up process. And in that step, you can expand the cells, you can activate the T cells, and you can transduct, and then you can have CAR T expression, and you can have all of these using, for example, Eppendorf solutions. And those are the solutions and controller that we have and we can provide. So in this case, treatments can be made in advance and will be available as soon as the patient is referred for treatment. And this is very convenient in cases where it is necessary to administer multiple doses because patients no longer respond to the initial treatment. Also, because this is off-the-shelf treatment, there is a minimal risk of manufacturing failures. And finally, I want to say that allogeneic product can be manufactured in batches, allowing for more efficient scale-up processes. For these reasons, there is a significant shift toward allogeneic therapy development. As you see in the slide, we see a big shift coming from autologous cell and gene therapy to allogeneic cell and gene therapy strategy. So according to the global market estimate, the allogeneic cell therapy market is expected to grow at the annual growth rate of 9.6% from 2022 to 2027, where the largest segment will be stem cells and also cancer therapies. Also, a CRB group serving more than 500 leaders in the life science industry, and they found that the developer from larger, smaller, and even CDMO organizations are moving away from the small patient-specific budgets and toward allogeneic processes. The immunological rejection is one of the principal disadvantages of this therapy. So I want to explain a little bit more here. So the host immune system attack the implant cells, causing side reactions to the patient. So they can cause between mild to severe side effect to the patients. So I don't want to go into detail here, but there are certain gene therapy strategies to prevent graft versus host disease. But the idea is to have or to use a universal allogenic cell source for different biomedical application. And I have to say that is one step closer to reality. In this sense, induced pluripotent stem cells, and everybody knows that IPSC, are derived from somatic cells, and they have been reprogrammed back into the embryonic live pluripotent state that allow for the development of any human cell type needed for therapeutic purposes. Yeah, to deliver off-the-shelf and inexpensive iPSC-derived products to more patients, it's necessary to generate a universal iPSC that can be transplanted regardless the human leukocyte antigen HLA allotypes. I will explain a little bit more what that means and what is important. So HLA are cell surface proteins, and if the T cells of the transplant cells and the immunohost system and they do not recognize each other because they don't match the HLA, there is an immune response, and then the tissue is rejected. Yeah, most of the strategy of HLA editing consists in two different steps, and I will explain them here. So first of all, the complete suppression of HLA to avoid immune rejection, and then we need to express certain molecules in the cells that we are implanting in order to avoid natural killer cells attack. So let me go into detail here. Let me explain more into details what is going on. As you see in the slide, in order to delete HLA expression, both HLA1 and HLA2 must be deleted. And but because of these regions, they are very large. It's very difficult to delete that by gene editing. So the easy way to suppress that is by deleting B2M, which is the common subunit of HLA1. So as you see in the slide, so if you want to delete HLA1 and avoid T cells attack, so we will delete the B2M portion, and then by doing that, you will delete completely the HLA1 class 1. Then we'll do the same with the HLA class 2 by deleting CIIT8 gene, and which is a transcription factor essential for HLA2 expression, and then we can suppress the HLA2 by doing that. The point is that if we lost HLA class 1, which is a ligand for natural killer inhibitor receptor, that situation will cause that the natural kill cells attack the new allogenic iPSC cells. In order to avoid that and escape from that situation, we have to force some expression of different inhibitor ligands like HLA-C and HLA-E, as you see here in the slide. This is a very complicated process, but I see some papers that people in the field working on that. So currently, there are many clinical trials involving allogeneic iPSC-derived cell therapy. For example, the iPSC-derived NK cells to treat solid and liquid tumor. Also, allogeneic iPSC derived cardiomyocytes in the treatment in patients with ischemic heart failure. Also, iPSC-derived mesenchymal stem cells for the treatment of steroid-resistant acute graft versus host diseases. And finally, iPSC-derived CAR NK cells to treat subjects with multiple myeloma. So the manufacturing process of the iPSC nowadays, including the preculture and reprogramming, is a labor-intensive and open procedure which need a lot of daily manipulation, including medium refreshment, and that can cause. And this is a high risk. This is a situation with high risk of contamination, and we should avoid that. So. And also, the scalability is very important for the allogeneic cell therapy development, but it can be difficult to achieve. The goal is to build a controlled manufacture process that reliably can produce the cell year and quality generated in the early development stages for clinical trials and commercial and commercial scale. In this sense, starting materials undergo an expansion process, and they are transferred into large-scale platform, such as bioreactors, which can be used subsequently to generate cell quantity required for allogeneic cell therapy. So we need to produce a lot of cells to be able to use them for many biomedical applications. So in this sense, it's important to track the IPSC morphology and also the aggregate size, because you don't want them to get and to grow really big, because you can get necrotic areas in the core of these aggregates and you can lose the cells. And also because the IPSC have a lot of spontaneous differentiation, and you don't want that situation happen. Also, the final dissociation process of the IPSC aggregate is a challenge because it may change the cell characteristic, impacting directly in the IPSC production. So we have an intense collaboration with Professor Robert Zweiger lab from Hanover Medical School. And during that collaboration, we have developed a new steering impeller design that support more homogeneous IPSC aggregate formation. And also we have developed a retention device, that is a kind of a filter device that allows keeping the aggregate of IPSC in this stereo suspension culture, allowing a continuous perfusion feeding. So without stopping the agitation, without stopping the gassing, we can continuously add medium and refreshing medium and into the system. Professor Zweiger laboratory has accumulated extensive knowledge over the years, with more than 50 publications in the last 3 years alone, as you see in the slide. So some of these publications describe various optimization studies, including agitation speed, pH, DO control, glucose supplementation, different feeding strategy, how the impeller geometry affects the IPSC proliferation and the aggregate formation, using specifically the DAXbox mini bioreactor system developed by our company. I want to highlight here that they have obtained 30 million viable cells per milliliter in a 150 milliliter working volume using this A-blade impeller configuration in perfusion. So now I'm going to introduce some details about what we have developed and the example that we developed in the bioreactor-based human IPSC aggregate production on a suspension cell culture platform using 1 liter bioreactor controlled by Cyberio twin bioreactor control system. So basically, as you see in the slide, how we were able to create the colonies in 2D, and by using Cyberio twin bioreactor control system, how we were able to obtain the human IPSC aggregate in 3D. So let me first introduce the Cyberio. So the Cyberio twin bioreactor control system is our newest controller. It is capable of controlling 2 single use of glass bioreactor, either individually or in parallel, and using a wide range of bioreactor size with a working volume from 1.3 to 40 liters, even though from microbial or for cell culture configuration. So we have performed 2 medium exchange batch culture, of course, using Cyberio and twin, and then we did that simultaneously using dasket vessels with a working volume of 0.8 liters and equipped with A-blade impeller optimized for human-induced pluripotent stem cells expansion. So let me explain in details the IPSC expansion workflow used in this work as you see in this slide. So the first step were the IPSC preculture, the colony picking process to avoid impurities, followed by IPSC characterization and to the expansion. So let me go and explain, the process step by step. So the experiments were performed using a human iPSC cell line derived from dermal fibroblasts obtained from a healthy donor, and we obtained that cell line from a TCC. So as you see in the slide, the iPSC were seeded in the vitronectin coat 6-well plate using a pro-warm stem flex medium from Thermo Fisher, also supplement with the Revitacell, also obtained from Thermo Fisher, at the density of 10 to the power of 4 cells per square centimeters, as you see in the slide. The medium was changed daily, and at day 5, you see a well-defined colonies, and this 6-well plate containing all these iPSC colonies was removed from the Cell Expert C170i incubator, and you see you have one of the picture in this slide, and place the 6-well plate in the laminar hood under the microscope for colony picking. So we did that, but if you see the morphology of the colony in this slide, you see that there are like they have a circular morphology. So we like trace a circle around the colony's border with a needle, and by using a P200 pipette, we remove the iPSC colony from the surface of the plate. This colony picking process has continued until enough iPSC cells have been collected, and then the iPSC cells were seeded in a vitronectin T75 flask and incubated at 37 Celsius degrees with CO2 percent of 5% of CO2 in the same incubator. So we have changed the medium the day after seeding, and then every other day until the cell reach the desired confluency. And we have continued this to the expansion process, as shown in this slide, until we obtain more than 800 million cells that they were the amount sufficient to prepare the inoculum for both bioreactors. Then the next step was the iPSC characterization through the iPSC pluripotency expression. So we have used flow cytometry, and also we have used immunocytochemistry. So let's explain a little bit. So the single cells that we collect from the T flask were incubated with conjugated antibodies against TRA160, against SSEA4 and 1, and also with the corresponding isotypes, and at 30 minutes at 4 Celsius degrees. So after that, the sample were acquired using a WAVA flow cytometer and analyzed by Flugio software. As you see in figure A, we show positive expression of this in pluripotency markers, and also this pluripotency was confirmed by the immunocytochemistry staining using O4 and TRA160 that you see in figure B. So after obtaining the amount of cells to prepare the inoculum for both bioreactors, the next step was the inoculation process and the iPSC expansion in suspension. So Before running any kind of experiment, so here, specifically in bioprocess, we have three important components to consider. First of all, the bioreactor, the controller, and then finally the inoculum. So let me explain all of them in detail. First of all, I would like to explain the bioreactor. So how we set up the bioreactor? The day before the inoculation, we perform, first of all, the pH sensor calibration by connecting the pH sensors to the Siberia Twin bioreactor control system. And then we perform this calibration according to the operation manual. So then each DAS give 1 liter glass vessel was assembled with the pH and DO sensors. Also, we have included 2 dick tubes along with the compression-proof adapter insert in a spare PG 13.5 port. So one for inoculation medium exchange, and the second one for inoculation and medium addition. Sorry. And also we have include 2 addition ports in each vessel head plate, one for base addition. In this case, we use sodium bicarbonate, and the second one for the addition of anti-foam, that in this case we use Pluripotent F60A surfactant. So after we assemble the bioreactor, we sterilize them and we put them, so we place them again back into their respective temperature control block to keep the system at constant temperature. So then we have connected drives, and also we have connected Peltier, exhaust condenser, and also the sparge line coming from the controller directly into the submerged sparge filters on the vessel. So once the bioreactors were settled and put in the heating block, we added finally the 600 milliliter of scale medium, supplemented with 0.1% of pluronic, and then 10 micromolar of Y27632 reagent, and conditioned for at least 24 hours using the parameters and subpoints that are listed here in this table. Finally, after 24 hours of conditioning, we inoculate the DASCAPE 1 liter glass vessel with 200 milliliter of medium containing 400 million cells, and with the density around 0.4 10 to the power of 6 cells per milliliter, with 97% of cell viability. So after that, the inoculation happened. So we waited like half an hour and we took sample from both bioreactors, and we did that also daily. And we measure also the pH and metabolites every day. So we did the measure of cell density and viability using a BioCell analyzer, and also the metabolite concentration was obtained using a CDEX bioanalyzer. So once we sample from the bioreactor, we have the IPSC aggregates and we have also the cell culture medium. So how we did that, we wait between 2 and 5 minutes in order for the IPSC aggregate to settle down in the bottle of our vial, and we took the supernatant, so that means the medium, to measure the metabolites. And then on the other hand, we use the aggregates to measure the cell viability and cell density. But of course, first of all we have to dissociate the cell pellet, and in order to do that, we use a gentle cells dissociation reagent from stem cell technology. So we add that for 5 minutes at 37 Celsius degrees and gentle pipette up and down, and we were able to dissociate the pellet and get the single cells. Then, as I explained before, that was a medium exchange performance, and we did that daily after 24 hours from the single cell inoculation until day 5. So how we did that? So we stopped the agitation and gas flow for 5 minutes, allowing again the IPSC aggregate to settle down in the bottle of our bioreactor, and then using a deep tube, we remove 40% of our cell culture medium from the top without disturbing the IPSC aggregates, and then using one of the liquid addition port that we have built in the head plate. Using that, we were able to include or add that again 40% needed with fresh medium without any supplementation. So as you see in the slide, we observe an initial lag phase, meaning that the cell viability decrease in day one. And we have been seeing this in similar studies. But as you see, that follow a rapid increase in both bioreactor between day 2 and day 5 of culture, reaching peak in viable cell density of around 8 million cells per milliliter, with a subsequent decrease in cell density and viability as expected. I want to highlight here that the cell density was higher than 90% up to until day 5. And also it's important to highlight that the high reproducibility obtained in this run from both bioreactor using our controller. And furthermore, we determine the consumption of glucose and the production of lactate and ammonia, as you see in Figure B, while at the same time we maintain the concentration of this metabolite below 2 grams per liter and 2 millimole per liter, respectively, during the whole run. So the downstream process of the IPSC expansion workflow includes the aggregate harvest and control test. And so we wanted to demonstrate that large-scale expansion process is compatible with the IPSC pluripotency, and that's why we want to study the pluripotency during the process, during the Scalable process. So first of all, in order to evaluate the growth, kinetic, and morphology of the IPSC under specific suspension culture conditions, the sample were harvested after 24 hours and after 24 hours of inoculation and every day until day 5. So how we did that? So we collect samples using a lab container bag with a line set, and Thermo Fisher provide that. And how we did that, we used that bag and we weld the bag into to one of our liquid addition port without interrupting the gassing and the stirring. So by doing that, we were able to collect our samples daily. And then using that samples, we obtained 10 bright field images from both bioreactors with mean average of around 20 single cells aggregate using images. So we use like 20 aggregates to collect all these pictures. And also we have monitored the human IPSC aggregates formation and diameter as you see in the slide. So 24 hours per single cell inoculation, the IPSC aggregate formation was found with the average diameter increasing for 95 microns in day 1, as you see in the slide, to 530 microns at day 5. I want to emphasize here that the ruler in the picture are equivalent to 400 microns. And we see like a totally narrow distribution of aggregate diameter in the first stage and in the first day, but after inoculation. But after that, we saw a broader aggregation diameter distribution at the late stage, as you see in the image. And finally, as I mentioned before, a large-scale production of human iPSC cells while maintaining the pluripotency is essential for clinical application. In this regard, most of the cell-forming aggregate harvest at day 4, because we did that specific assay on day 4. So, and we harvest this aggregate from both bioreactors. And I want to highlight here that the aggregate size was around 400 microns in diameter at that day. So all these samples, all these cells express pluripotent associated to surface markers like SEA4 and TRA160. And also we saw positive pluripotency in immunocytochemistry using transcription factors as such as OAT4 and nano-E, and similar to the same result we get when we have the human iPSC in monolayers. So I want to highlight that these beautiful images from our iPSC aggregates were obtained using a confocal microscope. So finally, we would like to announce our Stem Cell Community Day coming in November 21st in Leiden in Belgium. We really invite all of you to become a member of our community and connect with scientists from academia and industry to discuss the most recent advance and new technologies in the stem cell field. And with that, I would like to thank you for your attention, and I will be happy to take any question you have.
Thank you, Dr. Escobar, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just ask a question box located on the far left of your screen. Let's get started. The first question is there another strategy to avoid rejection of allogeneic grafts by the host's immune system after infusing iPSC cells?
Well, we have discussed here one strategy that is basically HLA editing iPSC cells, but of course, currently there are some strategies like immunosuppressive treatments. Like by taking a medication, you can, suppress your immune system. But the point is that basically take like 3 to 12 months for your immune system to recover from this transplant. And also I know another way. So by creating large iPSC bank for 100 donors that can match HLA allotype. So I recently read an article from Saira Foundation from the Kyoto University, and they have developed a clinical grade HLA allobank with human iPSC cells, and then matching approximately 40% of the Japanese population, something that is quite impressive.
Yeah. Well, thank you for that. The next question is, did you perform a karyotype analysis of the iPSCs that are part of the aggregates?
Oh, that's this is a good question. So as everybody knows, karyotype analysis is essential to determine any genomic alteration in the cells. But I have to say that we haven't performed that. We don't have data, but this is an ongoing study right now.
Sure, makes sense. Thank you. Our next question is, you mentioned that you used a 1-liter glass vessel fitted with 8 blade impellers. Have you tried other impeller geometry?
No, we haven't tried that. We have, as you see, I explained all these studies that Professor Robert Spiger have performed in his lab, and we kind of adapt the best conditions, the best possible condition for us to mimic or reproduce the iPSC aggregates formation in our laboratory. That's why we use a day-plane pellet configuration.
I see. Okay. Thank you. We have another question, which is, how do you recommend testing the quality of IPSC throughout the reprogramming and expansion steps?
Well, I have explained this in the presentation. There are many ways. So you can, for example, you can study the morphology. The morphology will change if something is wrong during the, your expansion. Also, you can use flow cytometry and immunocytochemistry to determine the pluripotency expression using, for example, what I did, DRA160, OG4, nano-E, etc. And but and also you can use karyotype analysis to see if there is any genomic alteration in the cells.
All right. Thank you for that. Have you ever tried any other pre-culture expansion protocol other than T-flask expansion?
This is a really good question. So this is an ongoing study right now. So rather than using or trying to expand the cells in 2D, we are right now trying to expand the cells in 3D using the aggregates in J-flask. And I think that we can save time and we can have more cells for the inoculation process.
Okay, all right. The next question is, I see that you did a medium supplementation. Can you explain a little more in detail?
So basically, we didn't do any supplementation. What we did was a medium exchange, as I explained before. So we have included a deep tube that was halfway in our bioreactor, and then because we need to do medium refreshment at a certain period of time, what we did we stopped the agitation and gassing, because if you have the agitation, of course, and gassing, all these IPSC aggregate will be floating in the medium. So we want them to settle down in the bottom of the bioreactor. And by doing that, we were able to remove the liquid from the top. And then by using one of the addition ports that we had already included in the configuration, we were able to include that 40% of fresh medium that we previously took out from the bioreactor. And this is the configuration we use to do medium refreshment in our workflow. Yeah.
Okay. Wonderful. Thank you, Dr. Escobar. We're going to wrap up with this final question. How do you digest the IPSC aggregates?
Well, also I mentioned that in the presentation. So that's it's pretty simple. Once you have the aggregates, so you have to remove the medium from that sample, and then you will wash the IPSC aggregate with the PBS. And then we tried different dissociation reagents, but we found that the gentle cell dissociation reagent from the stem cell technology was the best one. We obtained a high viability using that, and then we maintained the cell aggregate with 500 microliter of this reagent for 5 minutes under. In the incubator at 37 Celsius degrees, and after that we did up and down movement with the 1 milliliter serological pipette, because using tip you can damage the cells, and we found that by doing that we obtained the best results, the best possible results.
All right, excellent. Well, thank you again, Dr. Escobar, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, DASGIP, an Eppendorf company, for sponsoring today's webinar. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via a contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until April 5th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.